Question 4, here we go. This is a beautiful outline of you have got a school cafeteria and you've got some sort of food poisoning from there. It gives you a nice scenario here to go with this though. It tells you a lot about this bacteria. It tells you, test shows the bacteria responsible for the food poisoning was a Campylobacter, and it's a helical, it's not a rod, it's not a cone shape, it's a helical gram-negative bacterium. That is the information it gives you already as the very first go, and that colonizes the digestive tract, usually of birds, which means it comes in bird poo. So somehow the food prepared must have contained a little bit of bird poo, usually found on the outside of chicken eggs or some other poultry product. Okay, can also be in the meat. Then step 1 is literally look at your diagram. Which one has to be the Campylobacter in question? It cannot be a rod shape. It cannot be a cone shape. It has to be, because it says helical, so it can only be this one here or that one there. It can only be the spirally shape because it's helical, not rod, not cone. You also know that it's gram-negative. That's the one that hasn't kept the crystal violet purple staining. That's the one where actually the grams iodine and then the alcohol washed out the first stain, and the counter stain, the saffron in the red stain, has actually stained. So therefore the one that is stained red, that's the one that you need to highlight. So your answer is highlighting this one. Has been done pretty well, I have to say. Second question here, explain why this bacteria might be resistant to antibiotics treatment. You know it's gram-negative. What is it that a gram-negative has that a gram-positive doesn't have in its cell wall structure and outer structure? If I wanted to just quickly draw it up, if you've got a gram-positive and gram-negative cell wall, for a gram-positive you literally have a thick layer of peptidoglycan. With a gram, oh, that should be negative. Okay, a gram-negative, you have a fairly thin layer of peptidoglycan. Apologise, my terrible handwriting. However, you have got above it two separate layers, of lipids. Lipoproteins and liposaccharides, so lipid layers above it, which means if you've got penicillin, which actually stops enzymes that constantly, they're enzymes in your peptidoglycan cell wall which reattach, they're literally forming cross-links enzymes. Those enzymes constantly make cross-links happen, cross-links, so it's a polysaccharide structure here that keeps the strength up with the help of those enzymes. Penicillin stops those enzymes from working, which means gradually less cross-linking, the peptidoglycan is weakened, and bacteria cells will then burst, lysis. However, with the gram-negative, the lipid layers do not allow the penicillin to actually even penetrate, so the penicillin can't even get in, can therefore not interfere with the enzymes that cross-link and strengthen the peptidoglycan. Now the mark scheme is pretty nice with that one. Literally lipoprotein, lipopolysaccharides in the outer cell walls, and therefore we cannot actually have the penicillin penetrate further. So that is structure of the cell walls of prokaryotes. The next one here, let me get that to the top here. So you're incubating that bacteria that you have found, and you're incubating it specifically at 42 degrees, which is special for many birds. Many birds do have a higher body temperature compared to other not other mammals, because birds are not mammals, to other vertebrates. What does scream at me is you're given a number for birds, but it doesn't say a number for people. It doesn't say a number for people. So for goodness sake, give the number for people. You all know that human body temperature is around 37, 38-ish. So therefore, and I've had quite a few, if you give me the all the human enzymes would have been denatured, and human bacteria would have been denatured, therefore Not yet at 42. Denaturing happens in the region of about 60 degrees for human enzymes. It's far too quick. However, we're past the optimum for human enzymes. However, the enzymes inside the bacteria that obviously lives inside birds, that would have a different optimum. The optimum temperature there would be around the 42. Therefore, those bacteria would grow really, really well at the 42. Where are we? Most human pathogens would have an optimum temperature of about 37, 38. So therefore, only that one would grow well. The others wouldn't grow as well. Denatured is a bit strong in that context. Then comes your serial dilution question, and it's got a little booby trap hidden in here just because they're being lovely. They've hidden that number here, that with the micropipette you're actually only adding 0.5 centimeters square, so half a milliliter to the Petri dish, and obviously then spreading it and so on. So watch out for that later on, because that comes up in a question. If you see a little number that's kind of a bit sneakily hidden into it, that's the one that you need to look out for. So explain, this is 3 marks, so you need to have 3 different lines of arguments. Why do you use plate U? Why is U the good one? And why is it that you're not using V? And why is it that you're not using these ones here? Why is it that they're not being used at the top? Why is it that that one is rubbish? And why is it that that one is good? Okay, I've had a couple of you and I really felt like, hold on a second, I want to give you all 3 points, because you're exactly giving that actually we have got the correct number of colonies growing. So somewhere between 20 and 200 countable gives you a reliable estimate of what you have overall. And I would have said that that's exactly the right thing, but it's not the full story. You will need to also say rather than the other plates. So do say rather than the other plates. So what actually is it that's wrong with V? With V, way too few to give you an actual reliable guess, a reliable measure of what actually you would have overall once you are calculating away the dilutions. Too small of a count. And here, too large of a count. You could have clumping. You could have 15 colonies growing actually in one little heap. It's hard to identify the individual colonies. You could also, yeah, hold on a second, where are we? Plate U, not enough, sorry, U has got the right number of colonies, so you didn't even need to quote that it's between 20 and 200 to kind of get a good count. The higher numbers, you can't distinguish between the individual colonies. It might be clumped completely, and V has not got enough there to give a reliable estimate. Careful, you can't do accurate. Accurate means, can you accurately count to 3? Can you actually count the 3 little blobs that are in here? Okay, that would be accuracy of counting. It's a reliability thing, because if you only had that half a milliliter of solution squirted in here, you could accidentally have had a half a milliliter that was very low in bacteria or very high in bacteria, and therefore it doesn't give you a reliable estimate of the entire sample. Okie dokie, then a little bit of maths. Now it does tell you on the previous part for you, and that's a number that you're going to need to have, it gives you the 10,000 dilution factor, and it also hidden here told you that you have got only half a milliliter added. So what is it that you're doing in your maths? It tells you you've got your 69 colonies. Okay, you had diluted by 10,000. Can I put that side by side and it still shows? So you've got your 69. You're timesing that with your 10,000 colonies. You also need to make up for it only having been half a milliliter. So times that whole baby by 2, which I'm going to have to go through the mark scheme because I haven't got the calculator at hand. That gives you a horrible number, 1,380,000. Okay, so that's literally whack that stuff in the calculator. Now, it tells you to, does it do standard form, or what does it tell you to do? Show you've reached your answer, in the actual food, show you reacted, show how you reached your answer, estimated number of bacteria per centimeters. cube. No, that's per milliliter, basically centimeters cube. No, we have reached the end of it. I can accept both. I can accept you changing this into standard form. Mark scheme gives me both options. Okay, so the mark scheme gives me the option to now say, okay, that's already there. It gives me the mark. Okay, easiest to forget is the dilution factor times 2, because you only had half a milliliter. So easy to overlook. Last part of this, explain why it is likely to be an underestimate. There are two different versions of counting bacteria. You can do a viable and a non-viable count. I am sure that when Katie did this with you, you had the hemocytometer mentioned the idea that you actually can count what you can see in a little square, overlaying literally the bacterial culture onto a little grid. That hemocytometer is literally a little grid on a microscope slide, and count, literally physically count. So that would be including the non-living, but this time you've only had the living bacteria, only the viable count. Also, the degree of clumping is not clear. Clumping is when you're, instead of starting with one singular bacteria, maybe you had 2 or 3 at the same time starting to grow. That means you have got a colony that didn't just start with 1, but that started with 2 or 3, therefore underestimating how many bacteria you've got in there. So it doesn't include the ones that have recently just deceased, basically, in that context. Let's say you transfer them out of the micropipette, and at that point they kind of got scratched open and died. And obviously the clumping from not from a single bacterium itself. Right, that was question 4.